Can I just come in? Sorry, Dr. Lighten. This is our chair, Senator Barry O'Sullivan. Sorry, mate.
Hi. Hello, Dr. Lighten. So using that analogy where we've got, wild catch and we've got farm prawns, what you're suggesting here is that the scientific testing has concentrated on farmed carp, if you like, versus wild carp.
Yes, and if I can just elaborate on that slightly, the virus is very difficult to detect if it's present but not propagating disease. So really, in most settings, it's the diseased, dying fish which then are analysed by fish pathologists and they find the virus present. But if the virus is present but not causing illness and mortality, it's very difficult to detect.
No, but I
can elaborate a little further on that, perhaps.
Yes, of course.
So go ahead.
Yes, go ahead.
Hi. So in the UK, we've had outbreaks of koi herpes virus over the last 20 years or so. These generally occur in closed systems, so lakes which have been stocked with fish for the purpose of angling. Now, each year we get about 10 to 20 new outbreaks which are transmitted in various ways between sites each year. I think in the last 20 years we've had one outbreak on a river system which will have wild carp, high genetic diversity, which have escaped from various different localities. So that could be down to the riverine ecosystem, which is preventing the spread of the virus easily through the population. It may be that the population is dispersed over a large distance, or it may be down to the genetic diversity of the carp that are in those rivers, where we have a lot of mixing of individuals, so we have a higher genetic tolerance, so to speak, for the disease. There's more resistance. Now, in the Australian system, you also have a lot of genetic diversity in the carp. We don't specifically know about the immune genes, but overall the genetic diversity is quite large along the Murray-Darling system. You also have very high densities of carp there. So if the release is concentrated on portions where the individuals are grouping together in large concentrations, there would almost certainly be a large outbreak. Whether or not that is a good thing or a bad thing is another question.
Yeah, but coming back to what exercised my interest, and let me put this to you, Dr. Marshall, and you can tell me whether I interpreted what you said correctly. We're going through some baseline scientific work with respect to this, and it seems as though we've chosen cultured carp for the scientific work as a baseline, as opposed to using wild carp, where there could be some genetic disparity between the two, meaning that the results in the cultured carp we might not be able to rely upon them in terms of what's happening with the wild carp. No. Just a yes or no, because I'll abandon this if...
No, the testing that CSIRO undertook in Australia was on, as part of the looking at potential disease in other species. They used wild-caught carp from Australia for those tests.
Yeah, which would be the appropriate thing to do.
Yes.
Okay, sorry. Yes. I'm sorry, Glenn, I misunderstood that.
No, it's quite all right, because I've forgotten where I was. Oh, yes, unintended circumstances or impacts.
Well, I think there's two main uncertainties around this, and the first one is really obvious, which is the water quality impacts of decomposing carp. And the NCCP is currently running an anoxia program where they're modelling. They're using the biomass estimation from another project, and they're modelling what that death of carp might actually do to the water quality. But the bit that's missing is the biological response of the native ecosystems to that work. And
so Dr. Marshall, sorry, if you ever wanted to move home, I'll come in, pack it all up, load it, chuck it in a road train and get it to Darwin. I won't break one single thing. I don't understand this sort of science, so I really got to make this
Okay.
simple. So
So-
when So if the carp are infected, they die. Do they float to the surface? Do they go
Some
to the bottom?
float, some sink.
Okay.
They decompose. As they decompose, they strip all of the oxygen out of the water.
Right.
And in circumstances where there's not processes that put the oxygen back, like rapid flowing water puts the oxygen back, but if you're in an intermittent river where it's not flowing, there's no process to put the oxygen back other than diffusion, which is very slow. So the oxygen goes out of the water. There's models being developed to simulate that as part of the current program. But the assumptions around what that actually does to the native fish and the native invertebrates that live there are not currently being tested, and that's an area that I think is very important. Given the scale we're talking about, like the whole of the Murray-Darling Basin plus other catchments, the feasibility of a manual cleanup operation at that scale is just something that seems very unlikely to be successful.
Chair, for the time, I mean, I'm real happy to see, so everyone gets to go because we have someone else coming on the hookup too soon, I believe. But there's so many questions we need to ask.
I've already formed the view.
I
that
could just add quickly to that water quality issue, if allowed to. I mean, Faith will be able to give you some very good information on that. She's an expert on that particular aspect. One other additional thing to realise when these fish die, they profusely leak blood. So there'll be lots and lots of blood in the river as well. And Faith done some quick calculations, and she concluded, or she inferred, that the quality of the water of the Murray-Darling system, if this was to go to plan, would be comparable to sewage in terms of the levels of different nutrients in the river. So that's something also to bear in mind.
Well, while you're on that subject, could you just touch on the prospect of botulism and just see if you can, in a minute or two, tell us, I understand how botulism occurs, but can it then, if we had a flow event, do you know what the life cycle of around botulism is and whether it can travel 100 kilometres downstream? I mean,
I can't really comment on botulism, so it's outside my expertise.
Yeah, it's a little out of my expertise as well, but botulism occurs in the rotting fish. So wherever these are decomposing, there is high potential for it to occur, and that could be spread by birds as well and other wildlife that contract it, and this could be passed on through the food chain.
well, more importantly, if properties along there are relying of stock are naturally watering on the river, or estuaries off the river, or they pump the water out of the river to go up into to reticulate it around 100 troughs over the next 40 kilometres. If botulism has a life longer than, if it has a life that would allow it to remain active until it got into those farm systems, I mean, we'll lose hundreds of thousands of head of cattle if that happens.
Yes, I'm not sure of the life cycle of botulism, but as far as I'm aware, that could be it would have some life cycle outside of a decomposing fish. Some time period.
How do we want, just one, thank you for that, Mr. Lighten.